We're really pleased today to hear from Carolyn Bertozzi, and the title of her seminar is Cancer Immune Therapy Targeting the Tumor Glycocalyx. Carolyn is really one of the pioneering chemical biologists and pioneering glycobiologists. So without further ado, let me introduce you to tell us about your exciting science.
A whole bunch of seats in the front row, by the way, so I won't be offended if you come down. Thank you, Martha. When you're kind of like a local person and you get invited to give a seminar, you usually just assume you're going to give your talk and then that's it. But then the biology department, they were so kind as to say, well, would you like to meet with us during the day and have like a real schedule? And I was like, wow, that's really cool, So I learned a lot today from meeting with my biology colleagues, so thank you very much for making the time for that. And also, it's really wonderful for me to be a representative of this relatively new seminar program, which is our now annual joint chemistry-biology combined seminar, and I think I'm the second person to represent that. So that's exciting too, and I think we'll have a lot of fun doing this in the many years to come. We have a lot in common. For example, we're all enduring this interminable construction project right outside, but it's all good because it's going to be wonderful.
Fences down today in front of the...
Fences came down. That's great. That's kind of a step in the right direction. And then also, happy Halloween, right? Everyone has a plan tonight probably, and it's definitely the only day of the year I can get away with a jacket like this. I thought I would take the opportunity. So I thought I would also start with a Halloween-appropriate slide. But actually, this hopefully will have some meaning to you by the end of the lecture. And I think, all of us hope that when we give a lecture that people in the audience remember just one thing, and that would be enough. And that's more than most of us can say about lectures we listen to. So if you remember one thing, it's this. That's the thing. But it will have some meaning at the end, okay, hopefully. Okay, so I thought I would talk about the research that we do, which seeks to target what we call the cancer glycocalyx. And glycocalyx is a word that means the coating of complex carbohydrates that every cell enjoys. And they're basically, it's the totality of all the glycans that are attached to cell surface proteins and lipids. And I'll try to convince you that the cancer glycocalyx could be playing a role in the ability of cancer cells to evade recognition by the immune system. And conversely, we might be able to intervene in that process by targeting the glycocalyx with therapeutic modalities. Here at Stanford, most people, certainly in this audience of chemists and biologists, have heard about cancer immune therapy as an exciting new dimension in our ability to treat cancer. And you really can't go anywhere without gaining some exposure to the excitement surrounding this. So for example, if you read journals like Science and Nature, you'll see every now and then the cover dedicated to this subject. And I think Science, gave cancer immunotherapy the Breakthrough of the Year award, and I think it was 2014. It was a couple of years ago now, but it was in fairly recent history. And Nature's been talking about it. If you're at the bookstore, you might find books about it. When you're at the supermarket, stuck in line at Safeway, you might notice it's on the cover of Newsweek and the cover of Time. And I really love this cover because it kind of makes it sound like it's like an episode of Game of Thrones or something like that. The brutally selective, hugely expensive, life-saving trials of immunotherapy. Wow, who doesn't want to read that? That's pretty cool. And then, New Scientist. This is kind of provocative. So New Scientist went so far as to name immune therapy cancer's penicillin moment. Wow, right? I mean, if you think about what that implies. And then, of course, the cynic in me had to go on Google and Google cancer and penicillin moment, and it turns out cancer has had 5 penicillin moments in the last 20 years. Okay, so I don't know. So we'll see, right? We'll see. But it's certainly exciting times, and I think probably the person who did more than any of these outlets to popularize the notion of using the immune system to target cancer would be our own former president, Jimmy Carter. So you might remember a few years back, he had an awful diagnosis of metastatic melanoma in his brain. And at that time, or prior to that time, that was surely a death sentence. I mean, that was a horrible prognosis associated with that. But he was fortunate that, his diagnosis came at a time when he could go on one of the recently approved immune therapies. And he was lucky because he was one of the about 20% of patients with that diagnosis that actually shows a response. So, he may well have been cured, which is kind of like a retrieval from the grave, quite honestly, for people who would have that kind of a diagnosis. So that's when everyone started to pay attention in the public at large, and I think the excitement just continues to grow. Okay, so to frame what I'm going to talk about with the glycocalyx, I thought it might help to start by talking about the drug that Jimmy Carter took and how it works, okay, and other drugs in that same class that have come on the market more recently. And all of those drugs target a particular synapse between immune cells called T cells and cancer cells. And so T cells, like all of your immune cells, they travel around your body and they form synapses one at a time with the various cells they encounter, and they taste them and determine if they're good or bad. And if they're good, then they just move on to the next cell. But if they're bad, then they can react, and if they're really bad, they might kill that cell, okay? And that's what happens when a T cell encounters a cancer cell some of the time. And that's because the T cell will recruit to the synapse an activating receptor. For the T cell, it's the T cell receptor. And it can bind a complex that's on the surface of the tumor. And the details aren't important, but let's just say that that complex, it gives like a signal of badness. All right? And that activating signal can then basically cause that T cell to fire off. And if it's a killer T cell, it kills the target. But it turns out that T cells, they also have receptors, for example, one called PD-1, another called CTLA-4, that deliver inhibitory signals to the T cell. And they'll keep the T cell quiet. And they recognize ligands on a target cell, such as PD-L1, the ligand for PD-1, or B7, it's a ligand for CTLA-4. And if there's enough of these inhibitory signaling molecules that get recruited to the synapse, they can kind of overpower the activating signal coming from the T cell receptor. So the T cell is doing a calculation where it's looking for signs of badness, signs of goodness, which is what these ligands would be, and deciding how to react. And so it shouldn't surprise people to learn that a successful tumor often has evolved molecules that send the signal of goodness, of health. And that's why many tumors have been found to upregulate expression of PD-L1 or B7 or both, because that is a phenotype that is advantaged. Those cells are resistant to killing or recognition by the T cell. And so knowing all of that, people coming from the backgrounds of immunology and oncology figured out how to intervene, right? If you could block the signal between those negative or inhibitory signaling receptors and their ligands, then you could basically tilt the equation in favor of the activating receptor and allow it to deliver an activating signal. So the T cell will then kill. And so Merck was able to do that with the drug called Keytruda, which was developed under Peter Kim's watch. And that was the drug that Jimmy Carter took. Meanwhile, Bristol Myers Squibb came out with Yervoy, and both Keytruda and Yervoy are monoclonal antibodies that bind to these two inhibitory receptors and block them so that they cannot engage with their ligands PD-L1 or B7. And then BMS came out with Opdivo, which is very similar to Keytruda. It's a monoclonal against PD-1. And then just a few months ago, Genentech joined the party with their own antibody called Tecentriq, which targets PD-L1, but it has the same effect. Okay? So all of these antibodies are trying to achieve the same thing, which is to block interactions between inhibitory receptors and their tumor-associated ligands. Let me also take a minute to talk about what's happening inside the T cell. So at the biochemical level, how is it that the T cell receptor and molecules like PD-1 are fighting a battle over the T cell response? What's the calculation look like biochemically? So I borrowed this cartoon from a different review article, and now the topology is sort of inverted. So the tumor cell is on the top, the T cell is on the bottom. And so here's the T cell receptor, and it's engaging its ligand. It's an MHC peptide complex on the tumor cell. That's the activating receptor. And here's PD-1 on the T cell, that's the inhibitory receptor, with its ligand PD-L1. So now the calculation is occurring here at the cytosolic face of this synapse on the T cell side, because the activating receptor can recruit various kinases, and they're going to phosphorylate things. But meanwhile, this inhibitory receptor PD-1, it can recruit phosphatases. And the two that are kind of famous here are SHP1 and SHP2, the so-called SHIP phosphatases. And they are recruited by virtue of their ability to interact with an amino acid motif that's called the item domain. And that's a really important domain for immunologists, because it turns out there's a number of immune receptors that have these item domains. And when you see the item domain, it sets off a signal in your head that this could be an inhibitory receptor, because it can recruit the phosphatases. And as Martha taught me this morning, phosphatases and kinases are battling together. It's kind of a battle of who's going to get phosphorylated to what extent. Okay, so that's the level of the calculation. All right, so remember the item domain. So what does this have to do with glycosylation? Well, it turns out that there's a long history of literature that correlates altered glycosylation and cancer. And some of that literature includes things like poor clinical prognosis, right, or inability to respond to certain kinds of chemotherapeutics. And it's a really old literature. So, these papers were being published before I was born, which is like, 35 years ago or so. But anyways, it's been a long time. And it's a very frustrating body of literature to try and digest, because it's almost exclusively correlative with very little in the way of a mechanistic explanation. So we know that if you compare the signature of glycosylation from healthy cells and cancer cells, there's differences, and they're quite stereotyped for certain types of cancer. But what does that mean for the actual progression of disease? And what do those glycosylation signatures actually contribute to the process of cancer? That really hasn't been much to say in that regard. And just to give two examples of quite pervasive, widespread glycosignatures, I'll point to what we call hypersialylation and mucin overexpression. These are phenotypes that have been found on cancers from all different kinds of tissue origins, having totally different driver mutations. It's a common theme. And so hypersialylation, that just means an elevated density of glycans that terminate with this blue sugar, which is sialic acid. And it can be found in a variety of different contexts. And I've just pulled out some structures that are kind of famously associated with different malignancies. But if you kind of take a step back and add it all up, what this says is that identify cancerous tissue, it's highly likely that you'll find more copies of these structures than you would on the normal healthy counterpart. And the same goes for mucins. Mucins are densely glycosylated proteins. They tend to have this interesting rigid geometry. They're very long. And sometimes these phenotypes go together because the dense glycosylation on mucins might include sialylated glycans. So one way to achieve hypersialylation is to upregulate mucins that are sialylated. So this is sitting out there, and people like me who've been thinking about these things for a while, read these papers from time to time. And then about, I'm going to say like 10 years ago max, a picture started to emerge. And it came courtesy of the Human Genome Sequencing Project, like so many other things, and the identification of a family of receptors in humans that are now called the SIGLEC family. So SIGLEC is an acronym. It stands for sialic acid binding immunoglobulin superfamily lectin-like molecules. And there's 14 of them in human. And they all share a similar domain architecture. So they have this little crescent moon, that's the sialic acid binding domain. And then they have a various number of immunoglobulin repeats, a transmembrane domain, and then 9 of the 14 have that domain I pointed to on PD-1, the item domain. And the first of those that was characterized in any detail was originally called CD22. It's also SIGLEC2, and that is a receptor found on B cells. And it was shown by Paul Crocker and Jim Paulson, about 10 years ago at least now, that this receptor plays a role in suppressing the activation of B cells when it's recruited to the synapse with an antigenic target. In fact, if you knock out CD22 in the mouse, those animals are prone to autoimmune type diseases, right? We now know that virtually all classes of leukocytes, so every cell in your immune system, expresses Siglecs, sometimes more than one. For example, we often associate Siglec-5 with dendritic cells. NK cells have Siglec-7. Macrophages have Siglec-9. Eosinophils, Siglec-8, and so on and so forth. So these are widespread in the immune compartment, and they have the ability, through their ITIM domains, to recruit SHP phosphatases. And I just told you, hypersialylation is a common phenotype on malignancies. So if you put all that together, kind of begs the question, the answer to which I think is sort of hiding in plain sight, which is, do Siglecs function as checkpoint receptors in the context of tumor immune evasion? Or is the reason so many cancers are hypersialylated due to the fact that that could be a survival adaptation that protects them from immune surveillance, just like having PD-L1 would protect against T cell activity? So about 6 years ago, we started working on this, and we thought we would test this hypothesis experimentally, starting with a focus on NK cells, which are a type of white blood cell or leukocyte that is thought to be important in protecting us against cancer. And as I just said, NK cells have primarily Siglec-7, though a small population of them also have Siglec-9. Okay, so the NK cell, think of it kind of analogous to the T cell. Like the T cell, NK cells have activating receptors and inhibitory receptors, and they will form synapses with targets. Those receptors get engaged in some combination, and a calculation is performed. And if the NK cell gets more activating signal than inhibitory signal, it fires off. And what NK cells do is they degranulate, which means they kind of spit out little vesicles that have pore-forming proteins, poke holes in the target cell. Reactive oxygen species can be involved, as well as lytic enzymes, and they will basically just pop the cell that they're attached to. And we started by asking the basic question: if the tumor targets are hypersialylated, can they recruit Siglec-7 to the synapse? And if they recruit Siglec-7, can that protein recruit the SHP phosphatases to oppose the activating signal that the NK cell might be getting through its activating receptors? And can we test this experimentally? So then the question became, how can we control the sialylation status of tumor targets in an experimental setting? And to achieve that, we took advantage of some chemistry that we had developed, focusing on what we call glycopolymers that can insert into the plasma membrane. And in fact, I talked about these about a year ago in the context of a different project for anyone who happened to be there at that lecture. But just as a reminder, we had figured out how to chemically synthesize glycopolymers that look like mucins from a distance. Okay, and we anchor to one end a phospholipid tail so that they can insert into plasma membranes of cultured cells just by mass action. At the other end, we can put an optical probe so that we can quantitate their density on the surface of cells and monitor their abundance and their trafficking patterns. And by doing this, we can quite carefully modulate what kinds of sialysides and at what density are presented on the surface of a cell, and then try to correlate that with biological properties of that cell. So now, zooming in a little closer, natural mucins are polypeptides that are rich in serine and threonine residues, and those are the sites of attachment of the pendant glycans. And they always start with N-acetylgalactosamine that's in an alpha linkage. That's absolutely characteristic of mucins. Here's a little bit of structure just to kind of illustrate what these things look like in 3 dimensions. Because the sugars are spaced so close together on the backbone, they kind of force the polypeptide to stretch out just to minimize the steric congestion. And that also causes a rigidification of the overall structure. And we wanted to make molecules that look very much like these biological mucins, also at the same size scale as biological mucins. And early on, we didn't really have the means to do that with native polypeptide backbones. So instead what we did is simplified the synthetic problem by replacing the polypeptide with a very simple polyalkane backbone. And then that reduced the synthetic complexity to the point where all we had to do was synthesize something like polymethylvinyl ketone, just a functionalized polymer, where these ketone groups are spaced approximately the way that you see the sugars spaced in biological mucins, and then just click synthetic sugars onto those ketones using oxime-forming chemistry, which is mild, aqueous compatible, can be done on unprotected sugars with no problem. So then in the forward sense, for people in the audience who are interested in chemical synthesis, you start with MVK, and we did what's called a Raft Polymerization, which is a very nice method that allows you to very precisely control the length of those polymers and to modulate that as you wish. And it requires the generation of a Raft initiator, like this thiocarbonate, where we basically pre-installed the phospholipid tail and just grew the poly-MVK polymer right off of that lipid. Then we combine that polymer with glycans that have amino oxy groups. Those condense with the ketones and form the oximes, and at the same time, nucleophilically deprotect this sulfhydryl group, and that becomes a site to which we can add a fluorophore just using maleimide chemistry. And we characterize these polymers using all of the usual methods, but I think it's kind of fun to show a picture. And this is a TEM image showing the individual polymer molecules, and they look like little toothpicks, right? Little rigid rods, which is, you know, what natural mucins look like. So that was nice to see. Okay. And then we went ahead and built a collection of these glycopolymers in which we attached a variety of different sialylated glycan structures that were kind of inspired from the cancer literature. And in each case, these structures had an amino oxy group at the end. So this is the simplest thing of all, just nothing but the monosaccharide sialic acid. And down here are some more complex structures, like a tetrasaccharide called GD3, which has been found on renal cell carcinomas at high density, for example, and siala-Lewis X, which has been found at high density on leukemias and certain breast cancers, as well as a few structures with no sialic acid, and those are controls. Okay, so now this is how we do the experiments, and these are just cell culture experiments. We take a target cell type, and then we incubate it with glycopolymer of interest, and we just let those polymers insert themselves through the phospholipid into the plasma membrane, and then we wash away the unbound glycopolymers. And then we combine those target cells with purified NK cells from fresh blood. And we just rock the cells together, and they'll form synapses over the course of a few hours. And then depending on the information that's exchanged at the synapse, they might decide to kill the target or not. And then we just monitor basically the live cells and the dead cells. And although I show this cartoon as if it's like, you know, a bowling ball and a basketball, that's not at all what these synapses really look like. They're much more structured and complicated and beautiful, like this picture from Google Images, which I found. Okay. Okay, so what happened in these preliminary cytotoxicity assays? So what I'm showing you here is on the y-axis is the percent inhibition of cytotoxicity when you coat the cells with glycopolymers having these various different carbohydrate structures, all normalized for the same density of those structures on the surface of the target cell. And as a target, we chose Jurkat cells. It's a T-cell lymphoma that just so happens to have a defect in a glycosylation chaperone that results in a kind of under-sialylated phenotype. So in a way, the Jurkat cell I think of as kind of a null background that you can now add to with your own synthetic glycocalyx. So when the Jurkat cells have no polymer coating, there's a certain amount of killing by the NK cells. But when we coat those Jurkat cells with a sialylated glycopolymer, they're now somewhat protected from that killing. And that's true for virtually any glycopolymer we tested that has a sialylated glycan structure. It wasn't all that sensitive to the details under the sialic acid. But glycopolymers lacking sialic acid, which are these two, were not protective against NK cell killing. So that was an important piece of information, but it doesn't tell us whether Siglec-7, the receptor on the NK cell, has anything to do with this. And so to test the involvement of Siglec-7, we used function-blocking antibodies that bind to that receptor and prevent its interaction with carbohydrate ligands. So now you're looking at percent cytotoxicity it's kind of the inverse of the above graph. And so here are the Jurkat cell. This is like the percent of killing when the cells have no polymer coating or when they're coated with the sialylated polymer. So you coat them with sialylated polymer and they're protected. But if we now add the blocking antibody against Siglec-7, this jumps up so you lose that protection. Now they get killed again, even though they have the glycopolymer. And an antibody against Siglec-9 doesn't have much effect, and that Siglec is not found on most NK cells, nor does an isotype control antibody. So collectively, from these two data sets, we can say that the sialylated polymers protect the Jurkat cells from NK cell killing, and they do that in a Siglec-7 dependent manner. Now, according to our model, all of this would require the recruitment of Siglec-7 to the synapse with the Jurkat cell. So we wanted to look at that using microscopy. And here's a duplex between the Jurkat cell and the NK cell. And they don't look like much, but they really pack a punch. It's unbelievable to watch these things kill. And we can, using immunofluorescence, we can stain Siglec-7 and look at its distribution on the NK cell. And you can see it's quite localized at the synapse when the Jurkat cells are coated with a sialylated polymer. And this is just a false color overlay showing Siglec-7 immunofluorescence in red and the glycopolymers in green. But now, if the Jurkat cell had not been coated with a sialylated polymer and were not hypersialylated, Siglec-7 remains evenly distributed around the NK cell. It's not concentrated at the synapse, okay? All, fitting the model of recruitment of Siglec-7 as a protective mechanism that a cancer cell might use if it's hypersialylated. Now, one of the other things I like about the NK cell is they have a couple of different ways that they can kill. They are able to kill in an innate fashion, which is what these assays were testing, but they also interface with the adaptive immune response by virtue of having Fc receptors that can recognize the constant region of antibodies. So if your immune system generates antibodies against a tumor antigen, and those antibodies can coat the tumor in sufficient density, the NK cell can form a synapse with that target that's mediated through Fc gamma R3. And that is a very activating receptor. So a synapse of this type is very activating for the NK cell. They really fire off. And this is an important mechanism, not just presumably to help us protect our own bodies from cancer, but it's also important because that is a component of the activity of some very famous monoclonal antibody cancer drugs. And they act through this process which is called antibody-dependent cell cytotoxicity, or ADCC for short. So for example, Rituxan, or rituximab, that's an antibody that recognizes CD20, which is overexpressed on various B-cell lymphomas. And patients treated with this antibody, presumably the cells get coated, they get recognized by immune cells through Fc receptors, and that's part of the B-cell elimination process. Same with Erbitux, an antibody against epidermal growth factor receptor, and probably the most recognizable of all monoclonal antibody drugs would be Herceptin. That's a Genentech Roche drug that binds HER2, which is expressed on a majority of breast cancers and also on certain gastric cancers. And all three of these antibodies are relying in part on recruitment of immune cells through the Fc gamma interaction. And I was kind of surprised. I started reading about Herceptin because this is the one, if you recognize an antibody drug, it's Herceptin probably. You might know people have taken it, and it's kind of held up as a paragon of personalized medicine, right? Because the way that you clinically decide whether to prescribe Herceptin for a patient is based on the molecular marker, right? You look for HER2. But if you look at the actual numbers, Herceptin does not look like a very good drug, to be honest, because while it is true that 74% of breast cancer patient biopsies show HER2 positivity, a majority of patients who are HER2 positive don't actually respond to Herceptin. So the clinical decision making goes like this. A biopsy is taken from, let's say, a mass that's palpated or something observed in a mammogram, goes down to pathology, and tissue sections are stained with an antibody against HER2. And then the woman is scored as HER2 3+, 2+, 1+, or 0. And if she's HER2 3+, she's eligible for Herceptin because she has a chance of responding that's reasonable, which is still not that great. It's less than 50%, actually. But if she's 2+ or 1+, certainly 1+, they won't even bother giving her a prescription because the clinical data would suggest she has just a very low chance of responding, so it's not worth the risk. And 2+ is a gray zone that requires a secondary test, but at the end of the day, of those that are considered eligible, a small fraction actually respond to Herceptin as a single agent. Most HER2-positive breast cancer patients, which amounts to more than 80%, are either unresponsive or ineligible because their HER2 levels aren't high enough. So now it doesn't sound like such a great drug. And as you can imagine, with a drug like Herceptin that has a potentially enormous patient population but widespread resistance, there's been a lot of research trying to understand why don't patients respond. But I guarantee you could read those hundreds and hundreds of papers trying to understand mechanisms of resistance, and nowhere in there will you see anybody even mention the prospect that glycosylation could play a role. So, since so little was known about resistance, we thought it would be kind of worth looking at this. And so here's the question in cartoon form. If an NK cell can recognize a Herceptin-coded HER2-positive cancer cell and react to it, could you overcome that reactivity by hypersialylating the cancer cell? And I should point out that breast cancers are notoriously high in mucins and sialysides. Okay? And is it possible that those sialysides are recruiting Siglec-7 to the synapse, at least with an NK cell, bringing the SHP phosphatases, and can those phosphatases overcome the activating signals that are stimulated by Fc gamma R3? Okay? So we did assays similar to the ones that I previously showed, and here's the outcome. And in this particular graph, I'm showing an experiment with, it's actually not a breast cancer cell line, but a gastric cancer cell line, but HER2 positive. And when we combine these NCI N87 cells with purified NK cells in the presence of a HER2 antibody, this is Herceptin, there's a pretty nice killing response. Okay? ADCC works really well in these in vitro assays. But if those target tumor cells were hypersialylated with one of our glycopolymers, now you can see that that response is muted, and there's a lot less killing going on. And control polymers that don't bind Siglec-7 have no protective effect. And I won't go into all the details, this is all published work, but we kind of went through all the biochemistry to understand the signaling pathways, and the model fits exactly as we predicted. And the same phenomenon is observed with breast cancer cells that are HER2 positive, as well as Dowdy B-cell lymphoma, which is a CD20 positive lymphoma with Rituxan as the antibody. So I think it's quite a general phenomenon that hypersialylation could be a resistance mechanism against ADCC by Siglec recruitment. Now, if all of this is true in a clinical setting, I think the way that you would intervene is quite obvious because it's already been done for the T-cell checkpoint receptors. If it's true that Siglec-7 recruitment to the synapse down-modulates immune cell activity, well, you can just reverse that by blocking Siglec-7 with a monoclonal antibody, just like the anti-PD-1 antibodies that are already in clinical use. So we're in the process of making that kind of an antibody so we can test this experimentally. I think what's a little bit less obvious is, can you go after the ligand for the Siglecs the way that Genentech has done with Tecentriq, which targets PD-L1? And the reason I think it's less obvious how to do that is because we actually don't know the molecular identity of these ligands. So we know that they're sialylated because that's the minimum critical moiety that binds the Siglecs. But we don't know if this is one specific sialylated glycoprotein or if it's 100 different proteins that happen to all have common sugar structures. That we don't know. So it makes it hard to think about blocking a molecule with an antibody if you don't really know its molecular identity. But since we do know that the sialysides are important, we thought maybe we could target the ligand by simply cutting off its head with an enzyme called a sialidase. So we started making conjugates in which we took tumor-targeting antibodies like Herceptin, and fused them to enzymes that would cut off the sialysides, most importantly in the vicinity of the synapse between the two cells, and thereby prevent the recruitment of Siglec-7 and delivery of the inhibitory signal. Okay, so let's talk a little bit about sialidases. Lots of organisms make sialidases. Lots of bacteria make them. Viruses famously make them, like the influenza virus. Humans have them. All vertebrates have them. They're quite widespread in nature. And at the outset, we identified these 4 sialidases as interesting prospects because there was some literature about their structures. They had been expressed and studied biochemically, and we knew that they were quite broad-spectrum sialidases. It would take the sialic acid off of virtually anything that was underneath it. And historically, the first one we looked at was the Vibrio cholera enzyme because my postdoc, Han Shao, had developed a very productive expression system. So we just were able to get a lot of protein, and that made it easy to work with. And so the first target, this is a synthetic target, was this beast of a molecule, Herceptin sialidase version 1.0, which has a molecular weight of 313 kilodaltons. And that's because this is a rather large sialidase, and each one of these is 83 kilodaltons, and the antibody is 150. Okay, so this is a big molecule, version 1.0. We designed our fusion strategy around the idea of connecting the sialidase way down here near the C-terminus of the antibody heavy chain. And that's because our aspiration was that this molecule would not only target the enzymatic activity to the glycocalyx, but also recruit the NK cell Fc gamma R3 receptor at the same time. And it's known that the epitope that Fc gamma R3 binds to is kind of up here, despite my earlier cartoons. It's in the CH1 domain with a little bit in the hinge region. And so down here, it's about as far away as we could figure out how to position this enzyme to kind of keep it out of the way of that motif. So the way we connected the antibody and the sialidase was through a chemical fusion, which for us is actually easier to do than genetic fusions, believe it or not, you biologists. And we do that using a technology that dates back about a decade in my lab called the aldehyde tag. And so I won't go through the details, but the bottom line is if you clone this little motif, the CXPXR, into your protein of interest, and express it in cells, and they could be CHO cells, for example, or E. coli, in the presence of this enzyme FGE, then during the process of translation, that motif gets recognized, and the cysteine gets oxidized to create a new residue called formyl glycine, which has an aldehyde group. And the chemists in the audience will appreciate that an aldehyde is quite unique chemically in the context of a protein surface, and amenable to selective conjugation reactions like the oxime forming reaction I showed earlier on those MVK polymers. And so we bring along the cargo with an amino oxy group. It finds the aldehyde and forms a bond, and that allows us to site-specifically modify the protein of interest in a chemical way. And we had originally developed this to make antibody drug conjugates, and it's now, shameless plug, in the hands of a company called Catalent, and they rebranded it the Smart Tag. That's cool. But anyways, and it works really well for making antibody drug conjugates, so we figured why not use it to make an antibody enzyme conjugate. Okay, and this is what Han did. It was like the first thing he did when he joined my lab, which was just over a year ago. He took the antibody, in this case Herceptin, in which the aldehyde tag sequence was cloned near the C-terminus of the heavy chain, so it's got an aldehyde, and just condensed it with this little spacer group, which brings an azide to the party. And many of you will kind of think about what's going to happen next. Meanwhile, in parallel, he took that giant Vibrio cholera sialidase and did sort of messy, non-specific lysine modification chemistry with another linking group, a little maleimide, or NHS ester, acylation to put a cyclooctine. I would say statistically 1 per sialidase, but since it was a lysine modification chemistry, there's heterogeneity on this side. But no matter, these two will click together quite nicely and make this giant molecule, which he characterized using all the techniques you could imagine. And so now we had our first conjugate and could start asking questions like, does this molecule selectively desialylate HER2 positive cells? Because that's the goal, right? It's got Herceptin. So to ask that question, he started with a simple co-culture model where he mixed together HER2 negative cells with HER2 positive cells in the same tube and characterized their sialylation status and their HER2 status just with fluorescent labeling reagents. So here's a little field of cells in the fluorescence micrograph stained with DAPI. And there's a bunch of cells here, but you can't tell which is which, okay, from that picture. But here's the same field now stained with a lectin called SNA, which binds sialic acid. And you can see that all of the cells are sialylated. They're all green. But here's the HER2 channel. This is a fluorescent HER2 stain. These two are HER2 positive. So that's those two and those two, okay? And here's just the overlay. And then we can also analyze the population of cells more broadly by flow cytometry. So here's on the x-axis is the HER2 level. On the y-axis, you can think of this as the sialic acid level. So here's the HER2 negative cells, the HER2 positive cells, and they have the same approximate density of sialysides. So now Han added to the culture a relatively low dose of Herceptin sialidase version 1.0, okay? And now you can see here's a bunch of cells. And some of them are still sialylated, but you see these two, they went dark, and those are the HER2 positives, right? And over here in the flow data, you can see that the HER2 negative cells were, relatively unaffected in their sialylation status, but the HER2 positive cells dropped down into a lower quadrant. So it looks like at this dose, we have selective removal of the sialysides from the HER2 positive cells. But then, when Han increased the dose tenfold, all of a sudden he started seeing some off-target reactivity. So this is a dose where that proximity effect is sort of overwhelmed by having high concentrations in solution. At 60 nanomolar, we can just jump right over to here, you can see the HER2 positive cells are desialylated, but now the HER2 negatives are starting to drop down, right? So we're starting to lose the sialysides non-specifically at this dose, all right? So at the very least, this gave us kind of a range of doses to play with to then ask the next question, which is, if we can selectively desialylate the HER2 positive cells, does that have an effect on the killing potency of NK cells in the co-culture? So Han did a killing assay where he measured the cytotoxicity as a function of the concentration of either naked Herceptin or Herceptin sialidase conjugate, okay? And these are the target cells combined with NK cells, right, in the presence of these two drugs. And at first, he looked at cells which you would consider to be low HER2 expressors. They're both breast cancer cell lines, and let's call them HER2 1+, all right? So if this was a patient, this would be somebody who probably wouldn't respond to Herceptin and wouldn't get a prescription. And you can see that Herceptin has some kind of basal ability to elicit ADCC, but it's substantially potentiated when there's a sialidase on the protein. And same for both cell lines that are low in HER2. So that was pretty cool to see. And interestingly, and actually quite consistent with the model for how this all works, Han did a comparable experiment on HER2 high expressors. The SKBR3, that's a HER2 3+ cell line. And in this case, there's a lot of killing. Herceptin is just about really as potent as the sialidase conjugate. They're indistinguishable. So in other words, having a sialidase doesn't really do much for you when you're HER2 high, but it's quite helpful when you're HER2 low. And this is just a negative control. These are HER2- cells, and they're not affected by either one of these conjugates under these conditions. Okay. So another way to think about this is, remember, the synapse is a calculation because you've got activating signals and inhibitory signals, and it's the combination that leads to a decision, right? And under circumstances where cells are high in HER2, well, they bind a lot of Herceptin, and they're presenting a very strong activating signal through Fc gamma R3 that's concentrated at the synapse. You've got a lot of receptor, a lot of ligand. And that signal is probably so strong that it might not matter whether there's an inhibitory Siglec also recruited to the same synapse, because the negative signal can't overpower the strong positive signal. So these are the NK cells down here. So here, this would be a happy NK cell, leave the cell alone. This is an angry NK cell, going to kill, right? So a lot of this activating signal makes the NK cell want to kill. And you can remove, the Siglec signal by having a sialidase come to the synapse, but who cares? Because the seesaw is already tilted towards activation, so it just doesn't matter. And so that's what we think is the situation when you're high in HER2. By contrast, it's the people who are low in HER2 that could potentially really benefit from having a sialidase coming to the synapse. Because, if those people don't have a lot of activating signal through Fc gamma R3, they could be sitting over on the resting state because they have this inhibitory signal from the Siglec. And if you reduce the magnitude of that signal by having sialidase, the seesaw can tilt the other way, get the NK cell to activate. And so that's what we think is going on in this cell culture model. And now, of course, we want to take this into an in vivo setting and see if we can model this in an organism. But first, we have to get rid of that off-target activity, okay? And the problem, we're sure, has to do with the nature of this Vibrio cholera sialidase. So I mentioned we chose this up front because simply we had a lot of protein. But it's got significant liabilities. First of all, it's huge, 83 kilodaltons. But second of all, it's got this interesting structure where the catalytic core is in the middle. So that's the part that takes off the sialic acids. But it's got these two other domains, which are lectin domains. And the way this works, it's thought that it's like two hands kind of grabbing on to sugars on the cell surface and then a mouth in the middle that's kind of biting them off. So these lectin domains give this enzyme the ability to bind to the cell surface independent of the antibody, which is not what we want. And so when I look at this, again, in the spirit of Halloween, this kind of reminds me of Haunter, right? He's like my 8-year-old's favorite Pokemon, and Haunter, he's got a mouth and these two hands so he can hang on and chew, right? And we want to get rid of the two hands. We just want to have the mouth, okay? So, but again, go back and look at the other sialidases, and these other ones, if you look at these structures, they're all very similar, and they're all just like that catalytic core. So those are mouths, but no hands. So Han was now really invested in getting better expression systems for these, and he, together with Melissa Gray, who's a second-year student, managed to get their hands on a bucket load of the Salmonella sialidase. That's really great. So if that's Haunter, then this is Ghastly, right? It's Ghastly. This is like the predecessor. If you evolve him, did I get that right? I don't know. This is what it sounds like in my house every day. All right. Okay, so now the target, so this was version 1.0, right? Herceptin with these two big things here. Now version 2.0, Herceptin with these nice tight little proteins. Still a pretty big molecule, all told, but it lost, 100 kilodaltons. That's a good diet, I think, to go on. And Han and Melissa together built this molecule using a comparable approach, but now we thought we could do it a little more cleanly. So everything on this side is, pretty much the same, but over here, we decided we should do this site specifically, right, and have a kind of more uniform molecular composition. So Han and Melissa engineered a unique cysteine into this protein so that they could basically do a maleimide conjugation to bring a cyclooctine. Then these two things click together, and now we have Herceptin sialidase version 2.0, all right? And about 2 weeks ago, they sent me these really nice data, and I still haven't had time to do a pretty animation, but at least you can kind of get the point. Okay, so now these are the two molecules we're comparing, is 1.0 and 2.0, and this is, again, flow cytometry analysis where there's HER2 on the bottom axis and sialic acid on the vertical axis, so HER2 negative and HER2 positives. So now, version 1.0 is in the top row, and these are various doses. And you can see, just like before, version 1.0, at some point, you reach a dose where the HER2 negative cells start to lose their sialic acids. But this is version 2.0, okay? And go all the way, just go to the very end here. At this very high dose, there's essentially no desialylation of the HER2 negatives. And the HER2 positive lost all their sialicides way down here. So this is great. Okay, this is very low off-target activity. Now we have, I think, a much cleaner reagent to take into some more clinically relevant models, and that is a goal of the next year. And in the meantime, Melissa is also trying to make what I think would be kind of like the ultimate molecule, which is 3.0, which instead of being a chemical fusion between the two proteins, we would just make a genetic fusion, and instead of using a microbial sialidase, which would surely be immunogenic in humans, we would like to use a human sialidase. And there are 4 of them in human to choose from. And for logical reasons, Melissa has gone with Nu2, as it's called. And she's looking at constructs in which the human sialidase is linked either up here at the N terminus of the heavy chain or the C terminus of the heavy chain. And I should say to the biologists, genetic fusions are lovely, but you just have so few options. Whereas with a chemical fusion, you can stick it anywhere you want, really. So, there's my shameless plug. So we'll see how these things go. But right now, in the interest of time, I should just make sure that everybody gets thanked, because this was really a wonderful team effort that evolved over many years. And it all started, I guess you could say, with David Rabuka, who was an old Berkeley student who, when he left the lab, he started a company called Redwood, which was acquired by Catalent. And he was really big in pushing the aldehyde tag technology to the point where we could just use it like this. Then Camille Gaudula joined the lab, and he's a synthetic chemist, and he worked out that nice Raft Polymerization method to make those mucin mimetics, and he's now got a lab at UCSD. where he's still making glycopolymers and doing cool things with them. Jason Hudak, who was a grad student, who right now is a postdoc at Harvard Med School, and he worked very closely with Steve Canham, now a scientist at Novartis in Cambridge. And they did all of these early cell culture models with Siglec-7 killing and so on and so forth. And then these folks are in the lab, well, not right now because they're here in the audience, but if they weren't, they'd be in the lab. And Elliot, he's an MD PhD student who's done some really beautiful work with the sialidase conjugates together with Han. Han is a postdoc. He's on the job market right now. Many of you might have seen his applications. So I'll just leave it at that. And Melissa Gray, who's a second-year ChemH CBI student who's doing this beautiful work with the next generation conjugates. These are the people who did the funding. And now, after all that, do you understand this? That's all you have to remember, right? Cloaked in sialic acid. Nobody knows. You're a bad guy, right? Okay, and that's it. Thank you. 'Do you want to do questions?
Of course.
This face has been freaking me out the whole time. Nice costume, though.
Do you anticipate, with respect to kinetics, do you anticipate sort of a struggle where the cell adds the sialic acid back and the enzyme tries to chew it off? Have you looked at the kinetics and time courses of that?
Not really, honestly. I mean, so Han, I don't know, in passing, where is he? He's like out of here. Oh, I mean, I'm trying to think of the time. Most of your assays are like on the order of 4 hours to 5 hours, something like that. So I don't think we've probably let the whole system evolve long enough. Also with Herceptin, Herceptin's like an internalizing receptor. So everything we put on the surface at some point is going to go in, and there might be some recycling. It's a good question. Early on when we were talking about this, we were like, oh, all we need to do is get the sialic acids out of the synapse. That's all that matters. They can be everywhere else on the cell, who cares, but not at the synapse. And we were kind of surprised at how totally stripped these cells became of their sialicides. So there's, I think, a lot of mobility. And I don't really know what the longevity of these reagents would be, but that's a really good question. And I should say, before anybody asks me, because always somebody does, what about in a mouse? What about this? The problem in the mouse is that this pathway is not conserved in mice. Mice don't have Siglec-7. There's a lot of divergence in the Siglecs. So it's really hard to model this biology in the mouse. You have to engineer a mouse, and that's an ongoing thing.
You showed unequivocally, I think, that there's no question that glycocalyx is affecting the break. But is there any reason to think that mechanically it could have an effect on the activator, that it could influence the strength of that interaction?
Yeah, I don't know how much talking I should do here. So definitely, and I guess the short form is that mucins in particular have very interesting physical properties, and they definitely affect the bulk physical nature of the glycocalyx. They increase the stiffness and the thickness, and this in turn can lead to rearrangements of other receptors. And we've shown in totally unrelated work that, for example, you can affect the real estate covered by focal adhesions by simply putting mucins on the surface of cells. We had a collaboration with Manish Bhoot, actually, to look at the effects of these mucins on the T cell synapse. I wouldn't say we know what the outcome is, but it's entirely possible. Yeah, there could be physical effects that could oppose or reinforce some of these effects. Sharon, you're sitting right here.
So sialo ligands can be modified even further.
No, yeah.
Sulfation or whatever. Does that happen in the cancer cells? Does that affect the sialase?
Oh, I'll give you a big hug and a kiss, because now I can even talk more, right? So turns out, yeah, so sialic acid gets acetylated at the 9-hydroxy group, which I won't show you, but it's kind of dangling off the chain. And when it's acetylated, it's blocked. It doesn't bind Siglecs anymore. That's inhibitory. And there's an acetylase that puts it on and an esterase that takes it off. And there was a big study done at MGH a few years ago on genetic associations with autoimmune diseases, and the number one signal was that esterase. So I think the smart money says that there's a different dials on the rheostat here, and one of those dials is whether the sialic acid is acetylated or not. And those could be really interesting small molecule targets, I think, for immune intervention. Yeah.
I'm curious as to the model that is out there for the formation of the synapse, as you stimulate one of the receptors, either an activator or an inhibitor, does that recruit new proteins to the membrane, or take proteins already in the membrane and bring them closer to the synapse, or is that no?
Oh, Mark, you guys are sitting here as like a 1-2 punch in the front row. I got Irv Weissman and Mark Davis staring at me to make sure I get this right. But I don't know. From what we see, again, in the time courses that we've looked at, I think these are very mobile molecules, and there's no reason to invoke anything other than diffusive, thermodynamically driven recruitment to the synapse. Am I missing something here, or is it that simple?
Only that there's not one particular synapse. It's not a neuron. It can be anywhere on a cell. So that's why stripping the whole cell is important.
Yeah. Mark says it's important that we strip the whole cell, because the synapse is not like a neuron where it's like one point of contact. It's like more of a, it's like licking the whole cell, sort of. Yeah.
So when we were looking with anti-CD47 combining with rituximab, we put the tumor into mice that had no NK cells, but had macrophages and got the same effect.
Yeah.
So the question is, are you working with Siglecs that are on NK cells, or maybe those that are on macrophages?
Yeah, that comes up a lot. Okay, there's a couple of papers that would support, I think, the interpretation of macrophage Siglec 9 as being more important in some models than NK Siglec 7. About 20%, plus or minus 10, of our NK cells also have Siglec 9. So unfortunately, it's hard to separate out the 7 and 9 biology. In the mouse, of course, there is no Siglec 7, but they do have Siglec E, which is on macrophages. It's kind of related to Siglec 9. I think it's just going to be hard to model this in the mouse, and we'll see. But the nice, and I should also point out that all these immune cells, they have also Siglec 3 and sometimes Siglec 10, and there's usually multiple Siglecs. And then T cells induce Siglec 9 when they're exhausted, and things like that, they're changing. And so because of the complexity of this, not like PD-1, where when you hit PD-1, PD-1 is done, and when you hit PDL, you've hit PD-1 indirectly, and it's very linear. This is much more diffusive, but the sialicides are always required. So I kind of favor going after the sialic acid, because now it's Siglec agnostic. And it will affect macrophages and dendritic cells and T cells even. That's how I see it. But it's a good question about, should we have focused on NK cells? It's a little bit historical, the reasons that we did. Yeah?
So your approach to target tumors with this depends on the expression of tumor-specific antigens and availability of antibodies to those tumor-specific antigens. And assumption that the tumors express more, or their tumor cells are covered with more sialic acid than normal cells. If your antibody targets NK cells, the normal cells, and decylates those, where do you see specificity ratio of specificity and toxicity of this type of approach?
Was like the therapeutic window,
Yeah.
and do we worry about off-target effects and autoimmune side effects?
Yeah.
Yes, I think everyone worries about those things with any of these immune therapies. And what can I say? Without having clinical data, I don't think I can either defend or abdicate the approach, other than to say that it is clear that the sialic acid axis of immune modulation is quite significant for a number of tumors. And there's a lot of compelling circumstantial evidence, such as the high MUC1 phenotypes of many different types of malignancies, correlations of MUC16, which is also called CA125, with poor outcomes and immune suppression. And whether you can blame Siglec engagement for these effects, I think we'll only know in a human clinical setting. Because again, I don't think we can model this very accurately in the mouse. Yeah.
So following up from that, on the flip side, is there any genetic data that actually links defects in sial? I can't say that.
Yeah.
Sialylation
Yeah.
with autoimmune disease?
Yes, so there's this esterase story. So people who have a mutation that renders their sialic acid 9-O-acetyl esterase malfunctioning are prone to autoimmune disease, and this includes Crohn's, rheumatoid arthritis, psoriasis, some of the big ones. So that's out there. There's a null allele in Siglec 3, which is on microglia in the brain, which is protective of Alzheimer's. It's a pretty strong protective allele. And there's a whole rationale about, in the absence of Siglec-3, are the microglia more phagocytic than they might be otherwise, and how that could be beneficial, although somebody might say that could be detrimental. So yeah, there are genetic associations with Siglecs.
Cool.
And a total of maybe 5 labs in the world that work on them.
The human enzymes,
Yeah.
where do our cells normally, where are they?
Where do we make sialic
Right,
acids?
like in the lysosome? or
Okay,
something?
so yeah, well, so the way it works is we are in the cytosol and nucleus, we biosynthesize CMP sialic acid. And that's a donor for all of the sialicides. And then it gets pumped into the Golgi compartment through a transporter, and then there's 20 sialotransferases in the Golgi compartment that use it and put it on different things. Okay, and there's a lot of interesting genetic data around those sialotransferases too. They often come up in tumor transcriptomic signatures. Elevated sialotransferases is not uncommon. Yeah, there's a whole thing there. CJ.
And getting kind of, because I think you've unearthed some really cool basic immunology. And so getting...
You mean we?
Yeah.
Because he's in my lab. That was the best talk I've ever heard in my whole life.
Well, I guess I was just
What would you like to ask, CJ?
how could this pathway have evolved? Are there stages in development where hypersalivation is critical? Are there immunoprivileged sites in our body that need to be hypersilated? Have people even thought about this?
People have, like 3 or 4 people have thought about this. Ajit Varki has, certainly. There's a lot of theories about this, but, not a lot of experimental data. Yeah, I mean, so the brain is the place with the most sialic acid by far. No comparison. I mean, it's like the flux in sialic acid biosynthesis is unbelievable in the brain. There are sialicides in the brain that you don't find anywhere else in the mature adult, although some of them are developmentally regulated. Like polysialic acid is probably the most famous for this. Interestingly, if you go kind of evolutionarily into simple organisms, if you go to Drosophila in the fly, it's very interesting because whereas we have 20 sialotransferases, there's only 1 in a fly genome that's predictable. 1. And when you knock it out, it has been knocked out, there's serious neurological problems. 1 enzyme. And there's a guy who's at Texas A&M, where I happened to be last week, actually, who I caught up with, and he works on this, and he's been trying to find, like, what is sialylated by this 1 enzyme in the brain? And it's such a rare thing that you can't find it. It's needle in a haystack. So we're talking about how we could help and so on. But so it's interesting. So I think if you wanted to get to the kind of evolutionary origins of sialylation in the brain, you should look in Drosophila. I think that would be an interesting place to look. Oh, and that enzyme's expressed only in neurons in the fly, by the way. Transcript, at least the transcript. Yeah.
How far down the evolutionary tree do you have to find an organism that has Siglec-7 that's non-human, like the chimps or gorillas have?
No, they diverged even after chimps. Yeah, humans have more Siglecs than any other characterized mammal. And one of the thought processes is that they kind of have evolved as they're mostly involved in innate immunity, I guess. And innate immunity evolves in a way that's kind of together with the pathogens that you have to deal with in your world. And so, for example, there are a number of bacteria that have, during evolutionary history, figured out how to hijack the sialic acid biosynthetic machinery. They cover themselves with sialicides and hijack the Siglecs as a means to suppress immune recognition. So you get the feeling that there's a lot of back and forth between the pathogens and the Siglecs that might have been the drivers of that divergence. But yeah, there's no good animal model for the human Siglecs, at least the details of them. Although a few of them are conserved, like CD22 or Siglec2, the B cell one, that is actually conserved in the mouse. And Siglec3, CD33, is also in the mouse, pretty conserved. But then the rest of them are just off,
Just for fun, could you do a Siglec-7 column and see who ends up binding to?
Yeah. We and a few other people have tried things like that, and we're trying again right now as it happens. But one of the problems is that monomeric Siglec-7 doesn't have a very high affinity for anything, so it's not a good pull-down reagent. But you can oligomerize it and do crazy things like this. We have some other leads on how to find the real things that really bind Siglec-7. Which, if you invite me back again next year, I can talk about that. But yeah, I think, so far approaches like that haven't been very successful. but people are trying. Yeah.
Let's have one last question.
Hyperacylation is going to add a bunch of carboxylates to the outside. Does that do anything to the membrane potential of the cancer cells versus other cells?
You know, that's a good question. And people, since there's like 60 years of literature talking about relationships of hyperacylation and malignancies, embedded or along the way are these things like, oh, maybe it's like repulsion, electrostatic repulsion, and or something like that. Honestly, it's hard to imagine that affecting in a very broad way a cell's interaction with its environment, quite honestly, because there's counter ions everywhere. Although I do think local electric field effects could be important. And so there are some interesting stories about ion channels, which are sialylated, and if they lose their glycosylation, which then leads to loss of their sialylation, their conductivity changes. That's true for clotho. Clotho is a famously sialylated ion channel that if you point mutate the site of glycosylation, the whole thing gets screwed up. And the theory is an electrostatic. But that's a very local effect on specific molecules that have to do with ion conductance. And more broadly, it's hard to, I don't think it fits as well.
Let's thank Carolyn for